Welcome everyone to the part 3 of the workshop 16 of the UCLA QC Bio Collaboratory, Intro to Library Preparation for NGS. Today I'm pre-recording a lab session since due to this COVID-19 situation, we are not able to be present here in the lab to have hands-on experience. So I'm trying to do my best to explain all the details about the experiments that we're going to perform today. Hopefully you'll have a sense of what means to prepare a library for NGS. I'm actually next to the Agilent TapeStation, which is going to be our first topic today. And so let's get started. Okay, so as you can see, the Agilent TapeStation, we have this version, it's called 2200, which is not the newest version of the TapeStation that is available. There are a couple of newer versions of it, but that's what we have in the lab. It's composed mainly of 2 units, the instrument itself, which is the white block here, and also the associated laptop, which has all the softwares necessary for the run and the analysis part. Why we use TapeStation in the lab? Well, it's great for QC. It gives you the sizing information, the concentration, although we do not use the TapeStation for quantification. We prefer to use a Qubit, a soft fluorimetric approach for quantification. We believe it's much more accurate than TapeStation. And also for some application, for example, for genomic DNA and total RNA, it will give you a, let's say, quality score, or how they call it, integrity score, which gives you like the information about the integrity of your extracted nucleic acid, okay? There are several applications that you can run with the TapeStation. I think there's an extra application that you can run in newer models. However, these are the main applications that I see people using here in the lab. So we have a D1000, which is basically you have the information of your DNA up to 1 kb in length. And you have regular sensitivity and high sensitivity. When do we use this? We usually use this at the very end of the library preparation to check the final libraries, or at the very beginning of the library preparation in case we are dealing with a DNA sequencing experiment, to check, for example, the sonication, the fragmentation produced by sonication. Okay, so if we want to check whether genomic DNA has been fragmented correctly in the size range that we want, then we use the D1000 screen tape. Also, if you go on the bottom, you have another application. It's called D5000. So D5000, it's basically the same as D1000, but you can discriminate DNA that it's up to 5 kb in length. And this application requires a ladder. So if you think of the TapeStation as an automatic electrophoretic system, you basically perform the same basic steps as when you're performing electrophoresis, right? So you are loading your samples with a sample buffer, and then next to it, you're usually loading a ladder to just have the sizing information. However, for the D1000, there's an option to have a electronic ladder, so you have accurate sizing information without loading a ladder, which is very useful if you want to save some money on the TapeStation runs. However, for some application, for example, D5000, and we'll see in a moment the genomic DNA, you need to run the ladder every single time to have accurate information about the sizing. Okay, so I was talking about the D5000. So D5000 comes in a regular sensitivity and high sensitivity as well. And we usually use this to check for cDNA amplification. So sometimes when you are performing RNA library preparation, the first step instead of doing fragmentation, you do reverse transcription of the entire full-length transcript. And this is true, for example, for single-cell applications or for the SMART-seq type of approach. And so you have your fragments will be like an average of like 2.5, 3 kb, depending on how well the cDNA production and amplification went. And so that's why you need a bigger size than a D1000 for visualization. Another application that visualizes much bigger molecules, actually up to 60 kb, 60, it's the genomic DNA application. This is not very common, actually. I usually use it if I want to check whether my extracted genomic DNA has high molecular weight. And this is actually will become a very important assay to run, especially if you are transitioning to the third generation sequencing technologies such as TaqBio or Oxford Nanopore. We actually have a Oxford Nanopore right here next to the TapeStation. And because they usually are able to sequence longer molecules, you need to have the information about your original quality of the genomic DNA that has been extracted before performing the library preparation. Also, I like to use, it seems like very weird, but I like to use the genomic DNA screen tape when I'm processing cell-free DNA samples. And the reason why I do that is because sometimes when you are extracting cell-free DNA, which is a very short molecule, that it's usually between, let's say, 150 to 200 base pairs that it's free-floating in your blood. Sometimes you have, depending on who's extracting it, you have contamination from genomic DNA, so cellular DNA. And so if you have a huge contamination of genomic DNA, then maybe you want to rethink about your extraction method. Because what you're measuring, it's not only coming from the cell-free DNA portion, but also from the genomic or the cell-positive fraction of your extracted DNA, okay? Also, sorry, for the genomic DNA, it requires a ladder for each run for precise sizing, okay? On the bottom, it's the RNA application. It doesn't require a ladder, and it's still high sensitivity and regular sensitivity. We're going to see that. This is giving us the information about the integrity of the RNA. It's really important. So whenever you're extracting RNA, you can check the quality of your extracted total RNA, and that will determine what type of library preparation that you can perform, as you have seen in the previous, actually yesterday in class. We're going to see that the only difference between DNA and RNA is that the RNA, as you know, it's a single-stranded molecule, and it forms, secondary structures. And so the presence of secondary structure might interfere with the electrophoresis. And so normally you do electrophoresis in denaturing conditions. So you usually use chemicals that are poured into the gel, or you denature the RNA right before electrophoresis, but then you still need some denaturing agent during the run, because it takes quite a while to separate RNA. We're usually talking about like 30 to 60 minutes. While in the case of the TapeStation, every run it's usually really quick. We're talking about between like 2 to 5 minutes per sample, depending on the application. And so what we actually need to do is to denature the RNA at the very beginning, put them in ice quickly, so you basically don't give time to the RNA to just form secondary structures, and then load it directly onto the TapeStation for electrophoresis, okay? So what you actually need to run TapeStation, so you see like in this slide, a bunch of reagents. So each subtable lists you with a couple of numbers, the reagents that you need on top of your sample, of course, for each application. But at the bottom, you see like this orange table that lists basically the consumables, which are common to all application. You see the loading tips, which are specific for the TapeStation. And then you see the strip tubes and the caps, which are specific for the TapeStation if you want to run 16 samples at a time, or 16 or fewer samples at a time. If you want to run more than 16 samples at a time, we're going to use a different plate holder in the TapeStation, and you can load up to 96 samples. And so the 96 samples reagents are listed at the bottom of the orange table. But for today, we're just going to use the first three, so loading tips, strip tubes, and optical caps. Also today, we're going to do a RNA screen tape and a regular sensitivity, and a D1000 regular sensitivity for our final libraries. If you want to use the TapeStation, it's free to use for all users at UCLA. You need to receive training, formal training from me. It's a 5-minute training. Then you will be added to the Google Calendar, so you can just schedule your run. And so if you need some reagent catalog numbers, just write to me, and then I'll give you the list of all the reagents you can use on the TapeStation. Okay. Bottom line, what do you need? So you need tubes and caps at the top of the picture, tips on the left of the picture, screen tape, which is the most important thing, at the bottom of the picture. And then on the right, you'll see sample buffer and ladder, which I told you it's optional. So the two applications that we're going to see today, they do not require the ladder. So we're not going to use that today. Let's get to some pipetting. Okay, so as you can see here, I have all the reagents and consumables that I showed you before. So I have the screen tape. I have, for the sake of time, I already allocated my samples and the sample buffer, which is this one in the brown tube. And I have the tips for loading on the machine. And so we are basically ready to go. I've allocated 1 microliter of my RNA samples in here, and also 5 microliters of the sample buffer. That's what the RNA regular sensitivity protocol requires, okay? I actually have only five RNA samples. However, as you can see, I also have a 6th sample here, okay? So this is one of the few drawbacks of the TapeStation. You can only run an even number of samples every single time. If, like in my case, you have a odd number of samples, what you can do is you can just load water in the last tube. And so we can basically now have an even number of samples, okay? So I added 1 microliter of my RNA. I've added 5 microliters of my sample buffer, and then I've added 6 microliters of water in the last tube. It can be between 6 to whatever, 20 microliters, okay? It doesn't really matter as long as you have enough volume. And then what I'm gonna do is I'm actually gonna vortex for 2 seconds my samples. And then at this point, I'm just gonna spin it down, just collect all the liquid at the bottom of the tubes. And then, as I told you before, RNA sample needs to be denatured. So what we're gonna do is we're gonna put our samples in a thermocycler, okay? And then incubate for 3 minutes at 72 degrees to denature our RNA. Here we are, almost done. All right, so 3 minutes on. So I'm just gonna put my samples back in ice for 2 minutes. And while we are waiting, we can talk about the screen tapes, okay? So the screen tape is actually made of 16 little separate electrophoresis chambers. You can run as many as you want, up to 16, of course. However, if you are not running 16, like for example, I'm doing today, I'm just gonna run 5 plus 1, so 6. Remember, always an even number of samples. You can reuse the screen tape for the unused lanes, okay? So for example, this screen tape has been used in 6 lanes before. So what I'm gonna run today is an additional 6 lanes. So I have 6 plus 6, 12 lanes used. So I still have 4 lanes left. The screen tape and also the sample buffer are stored at 4 degrees, so in the fridge. And you usually take them out for about like 15 to 30 minutes before running those samples to just warm them at room temperature. The electrophoresis occurs at 25 degrees, so room temperature. So once you're done, I'm gonna show you later. I'm just gonna put it back in the foil and then put it back in the fridge for the next use. Agilent suggests you to use the screen tapes within 2 weeks of the first use. However, in our hands, especially for the regular sensitivity applications, even going after 2 weeks, usually they work just fine. For high sensitivity, you might have some problems, meaning that the information that is giving you is correct, but the information you might miss some lanes. So why don't we just go to the next to TapeStation, and we're going to load our samples and start the run. Okay, welcome back in front of the TapeStation. This is the closest look I can offer you of the TapeStation. It's basically composed of 3 main parts. I don't know if you can see this a little bit better. This part is the sample loading position. You have the tips, and also the tip holder is divided in 2 sub positions. The one on the left is the new tips that we're gonna put in, and the one here on the right is the reservoir that basically holds the tips that have been used already. Then we have to empty at the end of the run. And then here, there's a position to insert the screen tape, okay? So we're gonna do that. We're gonna put our samples in, we're gonna put our screen tape in, and then we're gonna put our tips in, okay? So in order to do that, let's get our samples that have been in ice for at least a couple of minutes. So the first thing that I usually do is I remove the caps. The TapeStation doesn't remove the caps for you. If you forget the caps, that's a $30K mistake. So please remove the caps before putting your samples in. There is a directionality, actually. This is a well A1, so sample number 1 usually goes here. 8, 9, 16, or again, A1, H1, A2, H2, okay? You can put the samples anywhere, even if you've used the tape already. It doesn't really matter where you put them, as long as you specify in your controller program, we're gonna see that in a minute, where your samples are, okay? So the machine will know, okay, so the samples are here. There are like these wells available. The machine will load them into the correct position, okay? So once I put in the my samples, I can put in my tape. You put in the tape in a specific orientation, okay? So if you look closely at the tape, you see a QR barcode on one side, and a green side, in this case, it's RNA screen tape. So the barcode needs to go on the bottom and then facing right, okay? So it's facing the laptop, okay? So I'm just gonna put this in like that, okay? And then we're gonna have some questions that the software is asking us, okay? We're gonna see that in a moment. For the tips, we are running only 6 samples. However, I usually don't care too much on how many samples I'm running, unless I'm running low on tips. I usually just put the full 16 tips into the system. And as you can see, I just drop the tips here. And the machine will only use the tips that are needed, okay? So the tips that are not needed, we can recover those and reuse them again, okay? So why don't we just switch to the other laptop for the software part, and then we'll come back here, all right? All right, so I am looking at one of the 2 softwares. So the TapeStation is controlled by 2 softwares. One that is controlling the run, which is called 2200 TapeStation Controller. The icon has the shape of the TapeStation. And the other one, it's called TapeStation Analysis, controlling everything that it's performed after the run. So if you want to modify some parameters, then you do that after the run, okay? So for now, we're just gonna open the TapeStation controller, which is basically controlling the run that we're gonna perform in a second. As you can see, the layout reflects exactly the layout of the TapeStation in itself. So on the left, you have the sample holder with 16 slots available. Again, I told you it's possible to load 96 well plates at the same time. In the middle, you have the tips holder. And then here in the center, you have the screen tape position, which is gonna populate as soon as I put the tape in the correct orientation, okay? I'm just gonna tell you right now, it's gonna give me an error because I opened this tape more than 2 weeks ago. So I'm just gonna click OK. So the first question that the TapeStation software is asking me is, what type of RNA are you running? Okay, so it's either eukaryotic RNA or prokaryotic RNA. And the reason the TapeStation is asking me that, it's because whenever we are running TapeStation for RNA integrity number, as you know from the lecture yesterday, the TapeStation is using ribosome RNA information. And as you well know, eukaryotic ribosomal RNAs are different from prokaryotic ribosomal RNAs, okay? So that's the only information that the TapeStation needs. So we're gonna select eukaryotic RNA, that's what we're gonna run today. So I'm gonna press OK. And as you can see, now this middle part populates, okay, with the information that it's green, it's RNA screen tape of regular sensitivity. As you notice, that you have like 6 lanes that are dark gray. So these have been used. So if you remember a few minutes ago, we talked about the tape that I was gonna use, and 6 of them were used. So that's an accurate confirmation of what I've written on the foil. And so, which means that you have 10 left lanes that are indicated as light gray in this screen. So, okay, so I've loaded all my samples. I've loaded all the tips. So actually I've loaded 16 tips, and I've loaded the tape. Okay, so now you have to select where your samples are. And we have positioned our samples in the first 6 slots. So I'm just gonna select the first 6 slots. Now this part on the right populates, okay? It's asking us, that's optional, okay? So you don't have to, but it's asking us to input the name of our samples, okay? And so I'm just gonna do 1, 2, 3, 4, 5, 6. Actually, the 6 is empty, so it's gonna be water, okay? And also, you might have noticed that as soon as I selected my 6 samples, 2 positions changed, okay? So these wells 6, 8, 9 through 12 got encircled in white. So these are the positions where the machine is looking for tips, okay? In our case, I put all 16, so I don't have to worry about specific positions. But if I were to run low on tips, I would have to precisely put the tip in those specific positions. The second part, it's these lanes that are, they turn basically from light gray into light green, okay? So those are the lanes that we are gonna use. You might notice that you have some, one green lane, one gray lane, and one green lane again. This is only a formatting problem. Well, it's not a problem, it's a different formats that the TapeStation is using. So if you notice on the left, you have a 96 well plate format, okay? So you have basically 1 through 12 and A through H type of format, while both the tips and the screen tape in the same space here, they don't have 8, they have 16. So it's more like a 384 type of format, okay? So that's just like formatting a difference between the sample and our screen tape, okay? So you don't have to worry about that. So once you're ready, you just double check again. You have samples in, no caps, just very important. You have tips. If you don't put tips, your samples will not be loaded into the machine, so you will have like all blanks. It happened to someone here already, okay? So just don't make that mistake. And then you have the tape in, okay? You put the description of your samples. Again, it's optional, it's not necessary, but it's always nice to remember what you put in. So you press start. As soon as you press start, it's actually asking you to save your run, okay? So we have a folder of RNA, and then you type in, so the actual information of the date, it's already present here. So we are gonna add test, run, a title, and then the extension, okay? So I'm just gonna click on save, and then the TapeStation is giving me another warning. Please ensure tips and samples are loaded. Close the lid and click OK to continue. To this one, I usually add, just make sure there are like no caps on top of the tips. Okay, so I have to close the TapeStation, and then I click OK to basically start the run. And then the run, it's currently going. I'm gonna pause the recording right now, and then we'll come back when the run is over. Okay, so we are back. The run finished, and the analysis software automatically popped up. These are our results. So you see samples 1 through 5 got something in it, while the last one, it's empty, it was just water. So what I'm gonna do is, as you can see, there's no sizing information, so I'm just gonna add a ladder. As you remember, RNA is one of the applications that doesn't require a ladder in the run itself. We could have done it since we only had like 5 samples, but I just wanna show you that you can get away with just using the electronic ladder, okay? So we're gonna insert the electronic ladder, so now you basically have these sizing information. The next thing that I usually do is I scale to sample. That's just like a visualization aid. So basically, it normalizes the concentrations among all samples. It's much easier to see features that will give you, just visualize the features. As you can see, samples A, B, are very good. So the RIN score, it's a number that goes between 1 and 10. And so these 2 samples have a RIN score of like 9.1 and 9.5, which is really, really high. You can definitely see the 2 bands of the ribosomal RNA bits, okay? Sample C, it's a sample that has, somehow good RNA, not perfect as sample A and B. I would be confident in doing a poly A capture using this sample. I might expect to have some 3' end bias on my final reads, but we can still do poly A capture on these samples if we're only interested in messenger RNA. While samples D1 and E1, you see also from the read score that the quality is really, really bad, okay? So we are going to take a different look at these samples. So this is like all the samples all together, just like a summary. And then we can switch to electropherogram, and then we have the visualization for each of the samples. The green bar that you see here at the bottom that every sample has, it's the lower marker, okay? The lower marker is part of the sample buffer, and it's like the regular electrophoresis, either the yellow or the blue dyes, depending on the sample buffer that you're using. It's running very, very fast. So it's the fastest of them all. And then it's setting the sizing information that the electronic ladder is using to get you the information of the size of these two peaks, okay? So this is basically the electropherogram of this sample number 1. And as you can see, you see the two prominent peaks of 18S and 28S eukaryotic ribosomal RNA. And then you see some noise in this region. And so basically here you have, it might be tRNAs and also the 5S ribosomal RNA. And all the noise, the background that you see here, it's basically messenger RNAs, okay? So you don't see a peak for messenger RNA because the size of messenger RNA, it's very variable, and the frequency of messenger RNAs is not really high. So we're talking about like usually 5%, okay, of the total RNA that you have loaded here, okay? So if we switch to B1, it's the same. C1, you start to see that the peak of the 28S is a little bit smaller than the 18S, and the ratio of the area is not as high as in sample A1 and B1, but it's actually closer to being 1 to 1, okay? So I think the perfect RNA would have twice as much of the area surface for 20S compared to 18S, okay? Then if you go down to samples D1 and E1, you see that you don't see those ribosomal RNA peaks anymore. And then everything is around like 200 base pairs, very degraded. Same for E1. Actually, the machine tried to assign this peak to 18S, but I don't believe it's true. So I usually go sample by sample to correct these errors. And as you can see, the read score changed a little bit, decreased a little bit. But again, here you don't see any huge peaks that can be identified as 18S. Okay. What else? There's also the DV200 method. So basically calculating how much of the sample it's above 200 base pairs. For this, we need to use a different function, which is the region table right here. Before going there, I just want to show you that the TapeStation is also giving you the concentration information for each sample. The concentration information for RNA from the TapeStation is quite accurate. So for RNA quantification, except for some samples that are really low in abundance, I consider it to be correct for all systems. So Qubit, Nanodrop, and the TapeStation, they're usually quite accurate. Okay. So you can use this information to quantify your total RNA before starting library preparation. Okay, so let's switch to the region table. So we can actually create two regions and see how much of the RNA sample it's above 200. To do that, again, we have to use the region table, but we have this file option that we can insert the regions. So as you can see, I already preset the two regions. So this is the DV200. And so it's going from 40 to 200 and 200 to 8,000. I'm not going to use 0 here because I have signal at 25 nucleotides, which is the lower marker. So if I do 0, then everything is going to change incorrectly. Okay, so I'm just going to apply to all my samples. And then I'm going to click OK. Where are you? File, options, regions, apply the file. Sorry, forgot to do that. Okay. And so now you see your sample that has these two regions, so like a dark green and the light green. And so for each region, it's telling you, okay, so it's going from 40 to 200, and then I have 37% coming from this region, and then from 200 to 8,000, so basically everything else, it's close to 60%, okay? I did prepare the libraries from these samples with the ribosomal depletion method. The results were not great. So since I had quite a bit of those samples, I re-prepared these samples using a targeted approach. So basically, I prepared libraries from total RNA and then use the hybridization capture to capture only the portions coming from exons, and I got much better results. Okay, and as you can see also here, this is slightly better. It's 65%, it's about 200 base pairs. In this case, for A, B, and C, so you see like C, it's like 83%, 78%, and 73%. When the RIN score is high, actually this DV200 does not make too much sense. So I wouldn't consider the DV200 in the case of A, B, C, just the RIN score, which is quite good. But in case of these very degraded samples, in order to understand whether it's good to go ahead and prepare a library or not, I will use the DV200, okay? All right, so there's two more things to talk about for the TapeStation, for the RNA application, and then we're gonna stop here. We're gonna create a report, okay? So you can create a report, and it's gonna be a Word document. So I'm just gonna save as, and then we can do it on the desktop, and save it as a Word document. So I'm just gonna save it here. And then we're gonna take a look at that. So we are all done here with our calculations and adjustments of settings. So we just close this one, save it, and where are you? You are here. So this is the report that's been created by the TapeStation. As you can see, you have page 1 with the summary information. So you have the gel image. You have the table with all the information about the RIN score, the 28S, 18S area, ratio area, and then the concentration, name of the samples. And then for each sample, you have one specific page. So in this case, you have the electronic ladder, which we didn't run, we just added after the run. And then you have sample A1 with the electropherogram, and then you have the information about the electropherogram. Then you have the region table, so you see like the dark green region and the light green region, and the information about the two regions that we've created, okay? And that's exactly the same for all the other samples. So sample number 2, sample number 3, sample number 4, sample number 5, and then the sample that it's empty, okay? One last thing, since this instrument is it's open to users, we have to clean up after ourselves, okay? So I'm just gonna stop sharing the TapeStation laptop, and then we're going back to the machine itself. So samples, we've already run the samples, so we can just stretch them. The screen tape, we still have some lanes available, so I'm just gonna place it back into my foil and then put it back at 4 degrees. The last part to clean up is actually the tips, okay? So I'm just gonna collect my tips on the left, okay? So the tips on the left are the ones that haven't been used, okay? The tips on the right, the ones that have been discarded. So I'm just gonna collect the tips on the left, and then I'm just gonna empty my bin, okay? I'm just gonna empty this one, and that's it. It's all clean, okay? So no samples in the sample slot, no tips whatsoever, anywhere, unused, used. Just make sure sometimes they get stuck. And then the screen tape port, it's empty. So we just close it, and then the next user will be good to go. Okay, can you hear me just fine? We're gonna stop here for 10-15 minutes. Let me check. And then I'm gonna answer, so please type in your questions. I'm gonna answer to your questions about like 2:30, 2:35. And then the class will start at 2:40, okay? So I'll see you at 2:30, 2:35 if you have questions. Otherwise, I'll see you at 2:40 back here, okay? I'm gonna pause the recording right now. Resume recording. Yeah. So it's divided in 5 parts. The first part is fragmentation and repair A tailing at the same time. The fragmentation is enzymatic, okay, so it's not sonication. And without purification, we proceed directly to adapter ligation, which is the second step. After the adapter ligation is over, we're going to perform an AMPure beads purification, a cleanup. After that, we're going to perform PCR, and then a final cleanup again. Okay, so you're going to see twice the AMPure beads purification with different settings, and all the other reactions are basically just enzymatic reactions, so they're very easy to understand. Okay. The video, it's quite long. It's going to take about like 50 minutes. Hopefully you can stay concentrated for that time, and then we're going to have a pause and for questions after the video, okay? So here are the components. I'm just going to briefly describe them. During the fragmentation and unpolishing, you just have an enzyme mix, okay? And also we are starting, so our starting DNA, it's a DNA from the phage lambda, okay? We are starting from 500 nanograms, and we're gonna have 2 libraries, okay? One of the library is gonna follow the standard protocol, and the other library is we're gonna push it to have basically to contain adapter dimers, okay? So we're gonna modify the adapter ligation step. We're gonna modify the cleanup steps, okay? And these modifications are done just to increase the amount of adapter dimers. Anyways, after the fragmentation, we're going to perform adapter ligation, then purification, PCR amplification, and then purification again. Okay, so these are the components that you will see in the video, and let's get started. Just a few seconds to open up the video. Just here. Where are we? Sorry, I'm trying to share. I don't see the share button here. 1 second. Hello everyone, welcome back to the lab. Today we're going to perform a library preparation. We are going to start with some DNA. So audio and video, okay. Can you hear and see just fine from the video? Okay, perfect. All right, I'll see you in 50 minutes. And in this case, we're going to use lambda phage DNA, which is approximately 50 kilobases in length. And we're using 500 nanograms per prep. As I told you before, there's going to be two different preparation, one which is the canonical, and the other one we are pushing the level preparation to contain adapter dimers and primer dimers, so you can see the size of the contaminants that you might have in the lab preparation. And I'm going to tell you how to remove those after we check the final TapeStation results. Okay, so I have everything with me. I have my pipettes, my thermocycler already set for the first step, and I have all my reagents. I have my reagents, so input DNA, lambda phage DNA. I have my enzymes and buffer for the first step, which is fragmentation and polishing at the same time. I have my reagents for the ligation step, so T4 DNA ligase and the ligase buffer, and also two different adapters. These adapters are 15 micromolar each, and we're going to tag each library differently. So this is a really important step. Every time you're preparing more than one sample at the same time, please use different adapters. So whenever you are sequencing those libraries in the same lane, then you can distinguish the reads coming from each library depending on the tag of the adapters, okay? And then we have the two reagents for the last step, which is the PCR amplification, okay? So we are basically ready to start, and let's get to some pipetting, okay? Okay, so let's add 2.5 microliters of the DNA fragmentation polishing buffer to each PCR tube. Go. And then we are adding 17.5 microliters of our diluted lambda phage DNA. Okay, so now we can mix and briefly spin down these two components. Briefly spin down to collect everything at the bottom of the tube. And then I'm adding the enzyme the mix of enzymes that is needed to perform the fragmentation and polishing at the same time. So it's 5 microliters of the enzyme. Go. So the two different reactions that we're going to perform today, actually they will have exactly the same enzymes and volumes. Only the purifications and the adapter ligation step are going to be different. Okay, so I'm just going to mix it again, just to mix the enzyme, which is a very viscous solution, and then spin it down and ready to incubate. Then press start. Okay, so the program, I don't know if you can see it, the program is basically 1 minute at 4 degrees, and then we have 15 minutes at 32 degrees. And in that step, we're going to have fragmentation and most likely end repair and 5' end phosphorylation. And then at 65 degrees, we are going to have A-tailing, okay? So the product of this step is basically fragment and repair, phosphorylate, and then an A-tail to our products, and then we are ready for adapter ligation, okay? So it's gonna take about 50 minutes. I just wanna mention that the time at 32 degrees has been optimized for the size range that we are interested in. So we're aiming at around 300 base pair of average size of our fragments. but you can increase it or decrease it depending on the concentration, the size of the library that you want. So there's a lot of optimization that you can do there. But I've done this many times, and I found that 15 minutes at 32 degrees is what we are aiming to. Okay. And so with this, we'll come back when the program is over. All right, so right after we started the program for end polishing and A-tailing, I usually remove my AMPure beads, which are the beads that we are going to need for purification of the adapter ligated and prepped libraries. And they are usually stored at 4 degrees if we're talking about AMPure beads or similar. There's also another version, it's called spry beads. They're usually kept at room temperature. So the only thing that you have to do for the spry beads is to resuspend them. I usually just briefly vortex them and then and keep them in rotation. If you have AMPure beads that are stored at 4 degrees, there are like 2 things that you have to do. You have to warm them up at room temperature, and also you have to resuspend them, okay? So I usually take them out, briefly vortex them to resuspend them quickly, and then I usually transfer them into 2 ml tubes if I haven't done that already, and keep them rotating so they're resuspended and they go at room temperature at the same time. Okay, so here we are. Our reaction is almost done. Again, this is going to perform end polishing and then A-tailing of our DNA on top of the fragmentation, okay? This is an enzymatic fragmentation type of lab preparation. So I'm collecting my samples, and I'm just gonna actually spin them down really quickly. Just collect all the liquid at the bottom of the tubes. And then I'm gonna set up the adapter ligation, okay? So the adapter ligation step requires buffer, an enzyme, and the adapters themselves, okay? So how this reaction works is I'm adding the adapters first every single time, just because I prefer to have the adapters mixed with the end repair DNA before adding the ligase. Otherwise, ligase will start producing adapter dimers if you prepare a premix with the buffer, ligase, and the adapters. So the first thing that I do is I add the adapters to my library, and then I take notes of what adapters I use for each sample. So in this case, adapter 9, I'm gonna add it to my library A, and adapter 10, I'm gonna add it to my library B. However, if you remember, I told you that one of the 2 libraries that we are pushing one of the 2 libraries to have adapter dimers. So in this case, instead of adding 2.5 microliters of adapters to my sample, which I just did for sample A, I'm adding 10 microliters. So 4 times more the amount of the adapters in this case. Okay, so I'm pipetting 10 microliters, and then I'm just gonna put them into library B or library 2, whatever you wanna call it, okay? And I take notes of which one is which, okay? So sample A, barcode 9, sample B, barcode 10, okay? Once I have my adapters mixed into my samples, I am adding the ligation buffer, so it's 10 microliters of the ligation buffer. Also, this buffer is very viscous, okay? So please pipette it slowly, otherwise you're not gonna pipette an accurate volume. After I've added the buffer, I can add the water for sample A, which has 7.5 microliters less in terms of volume. And then I'm adding 5 microliters of my ligase into each sample. Also, the ligase, like every enzyme, is very viscous, so please So now I can mix my samples. Okay, so I just want to recapitulate what we've done so far. We have done fragmentation and polishing and A-tailing at the same time in step 1. Now the entire product without purification, remember these are streamlined protocol, are basically used as input for our ligation. So we've added adapters first, and then the buffer, and then the ligase at the end, mixed everything, spoon down, and then put in the thermocycler for 20 minutes at 20 degrees, and then we'll come back with the purification step using the AMPure beads at the end of the ligation. All right, so here we are again. The ligation just ended. So I'm just gonna take my tubes out. You see that the temperature is going down to 12 degrees. Also before it was 20 degrees, so it's colder than room temperature. So what we're going to do is we're going to incubate our samples at room temperature for a couple of minutes just to have them at room temperature, because all the steps that we're going to perform, so the library purification will be performed at room temperature. In the meantime, I'm going to briefly talk about the tubes that we are going to perform our purification in. As of now, we have our samples into PCR tubes. You can perform the purification in PCR tubes. Just use one of these or a similar magnet that fits PCR tubes. However, today, to let you see a little bit better, even if I know that the quality of the recording is not going to be great, I'm going to use 1.5 ml tubes. So what I'm going to do is I'm going to transfer our library that has been adapter ligated into 1.5 ml tubes. Okay, so what do we need for AMPure beads purification? We need our DNA. In this case, I have it in 1.5 ml tubes. Just transferred it from the PCR tubes. We need the beads, AMPure beads. They were rotating at room temperature since the first incubation. So they are very well mixed and at room temperature. And also freshly prepared ethanol 80%. Now, the two purifications are going to be different, as well as the previous step, the adapter ligation. If you remember, the previous step that we just finished up had sample number 1 with the standard library preparation requirements, which were 2.5 microliters of 15 micromolar adapters, while sample number 2, or sample B, has used 10 microliters of 15 micromolar adapters. So we basically pushed the formation of adapter dimers. Also, in this step right here, we are pushing for adapter dimer retainment. So they are co-purified. The adapter dimers will be co-purified with our libraries. Okay. The standard library requires 40 microliters of AMPure beads. So I'm just going to pipette 40 microliters of AMPure beads into my library. The AMPure beads, if you remember from day 1, they are very viscous because they are 2.5 molar of sodium chloride and 20% of PEG. So they're very, very viscous. So I'm just adding the AMPure beads to my solution, and then I'm mixing it. The other sample instead does not receive 40 microliters. It receives much more, 90 microliters of AMPure beads. And then during the incubation, we can talk about why we're doing this, okay? So I'm just gonna add 90, much more, and then pipette that in and mix it using the Vortex. Okay, so now we can start our 5-minute incubation for the beads to bind to the DNA. Why do we have these two different volumes of beads added to these two different samples? So sample 1, or sample A, which has the standard volumes, it's 0.8x purification. That's how we call it. So if you make the math, it's 40 microliters in 50 microliters of solution. So 40 microliters of AMPure beads, 0.8 to 1, which is 50 microliters of our adapter ligated library, okay? While the other sample, sample 2 or sample B, we are using 90 microliters of AMPure beads to 50 microliters of our library sample. This is a ratio of 1.8 to 1, or you can call it 1.8x, okay? If you remember how the AMPure beads work, the ratio of AMPure beads, I'm considering AMPure beads, not just the magnetic beads, but also the buffer that the beads are resuspended in, the ratio of this mixture to our DNA can determine the size of the DNA that is bound by the beads, okay? So the higher the ratio, the smaller the size of the DNA that the beads can bind, okay? So if we have a bigger ratio, like for example, for sample B or sample 2, 1.8x, also the smaller sizes of DNA will be bound, which means that also the adapter dimers and the primer dimers will be bound by the beads. But in case of the regular sample, we are using 0.8 volumes of beads to our DNA sample, which means that only DNA fragments of usually about like 200 base pairs are bound to the beads, and everything else is discarded. And we are gonna remove it during our washes, okay? So this is just the background idea of how to use the AMPure beads that you've seen also in day one, but now we're talking about the actual volumes that we are using here in the lab, okay? So all right, so as you can see here, sample 1, sample 2, there are two differences, the volume difference. In this case, we have 40 plus 50, so around 90 microliters of AMPure beads plus our DNA library mix. And in this case, we have 90 plus 50, which is about 140 microliters of mixed DNA library and AMPure beads. You also notice a color difference, okay? So sample number 2, of course, the more the beads, the darker the color. And sample A or sample 1, it's lighter, okay? Okay, so the incubation is almost over. What I usually do is I spin them down to collect everything at the bottom of the tube, and then we can put them on the magnet. So now we're incubating our samples on the magnet, and it's gonna take around 4 to 5 minutes for the separation to occur. All right, so our incubation is almost over. I'm trying to show you, so the magnet is here at the bottom here. And as you can see, hopefully, is that you see a bead pellet facing the bottom part, so where the magnet is, which is dark brown here and there. And everything else, so the rest of the solution is clear, okay? So if you look at from the front is less clear because everything is basically colored dark brown. But again, if you go close, you may be able to see that the pellet is separated from the solution, okay? So now the beads are pelleted against the magnet. What we can do is we are able to remove the supernatant, okay? One quick note about the removal of the supernatant now is that the beads are, yes, pelleted against the magnet. However, they're not strongly pelleted against the magnet. So if you were to remove the supernatant with the, P1000 pipette tip set to 300 microliters, you might remove the pellet. You might get the pellet during your supernatant removal. So it's really, really important now to know what is the volume of the supernatant you have to remove. And I usually prefer to leave 5 to 10 microliters of the supernatant in my tube, not to dislodge the bead pellet. And I'm not discarding the beads, which as of now attached the library DNA. So in case of sample 1, we have 40 plus 50, 90 microliters. In case of sample 2, we have 50 plus 90, so 140 microliters. What I'm gonna pipette is 85 microliters here and 133 microliters here, okay? Okay, let's try to do a close-up. So I am basically I'm setting my pipette to 133 microliters. So I'm hopefully leaving 7 microliters in without touching the bead pellet. So I'm basically facing my tip in the opposite direction as the magnet, and then I'm going in and then removing slowly my supernatant, okay? So let's check that the bead pellet is still there. There are still few microliters. I know you cannot see it, but trust me, you still have few microliters in the tube. And also what I usually like to check is whether my supernatant has a brownish color at the tip. That means that I picked up some beads. In this case, it's totally clear, so I'm good to go. I can discard the supernatant. All right, setting my pipette to 85. I'm repeating exactly the same thing, facing away from the magnet, which is the same as saying facing away from the bead pellet. I'm just going in, removing the super slowly, and then coming out. Again, few microliters left in my tube, and then checking the supernatant. It's clear, so I can discard it. So after discarding the supernatant, I can wash my bead pellet. We do this by keeping our tubes on the magnet. The volume of the ethanol that we're using for washing has to be enough to cover entirely the bead pellet, okay? So usually in 1.5 ml tubes, 300 microliters of ethanol, 80% are usually sufficient. Okay, so I'm also pipetting away from the bead pellet. I don't want to dislodge it. Okay, 1 and 2. And as you can see, the bead pellets are still there. Okay, and the ethanol is here. So we incubate for about like 30 seconds, and then we are able to remove the supernatant. One thing that is different between the supernatant removal of our first step compared to this one is that now when the ethanol 80% is there, the bead pellet is strongly attached to the magnet, or it's more, you can also think of it as the bead pellet does not resuspend into the ethanol 80% as easily as in the previous supernatant, okay? So in this case, what I like to use is actually a vacuum aspirator. So it's much stronger, it's cleaner. Here you go, a closer look. So I'm pushing my tip against the opposite side of the tube. It's still a supernatant removal, so I don't wanna touch my bead pellet. And then I'm going in and I'm removing my supernatant, okay? I'm changing the tip. I am removing the second supernatant. So same again, tube on the magnet, tip facing the opposite side of the bead pellet, pushing down and removing my ethanol supernatant, okay? And now we basically have dry tubes, okay? So there's a second step of ethanol wash. So we're gonna re-add 300 microliters of our ethanol 80% to each tube. Again, pipetting on the opposite side, pipetting on the opposite side, and there you go. And for now, we're done with the ethanol 80%. So we wait about like 30 seconds, and then we are good to remove the ethanol supernatant, the second ethanol supernatant. As you can see, the volume of the ethanol washes are much bigger than the original supernatant that we left in, about like 5 to 10 microliters. So I'm pretty confident that both washes of ethanol removed everything that we had left in the after the first removal of the supernatant, okay? Okay, so now we can start our drying time. I usually prefer to give it a couple of minutes and then going back in with my vacuum aspirator to remove all the droplets of ethanol that I wasn't able to remove from my ethanol removal. And then I would say usually 5 more minutes to dry our bead pellet. The drying time greatly depends on the type of tubes that you have and the amount of beads that you have that you want to dry. So I would suspect that the sample number 2 or sample B, which has more beads, will take longer to dry than sample 1. And also, if you were to perform the purification in PCR tubes, usually it's faster than 1.5 ml tubes. Okay, so let's wait 1 more minute. Why do I want to wait a couple minutes? So the vacuum aspirator is really fast in removing the ethanol. So it's possible that some ethanol that is still on the walls of the tube slowly comes down on the bottom of the tube. So that's why I usually want to wait a couple of minutes. And then I'm not only going on the bottom of the tube with my vacuum aspirator to remove everything else, I'm also looking for some other droplets that are on the walls of the tube as contaminant. All right, so time is almost over. So I'm going on the opposite side, same as before. Go all the way to the bottom. And then I'm looking for droplets that are here around. It's pretty clear. And also here, the other one. All right, everything is fine. Okay, so let's give it about, let's say 3-4 minutes, and then we are gonna check it again. The drying time of the beads is really important for 2 different reasons. First of all, you want to remove as much ethanol as possible, not to have some ethanol carry over in your next reaction, which will inhibit it. And also, you don't want to over dry the beads. If you over dry the beads, the resuspension with the elution buffer, that it's going to be the next step, is not going to be as efficient as if they are correctly dried. Okay, so you just want to get the timing right. And you can see that. So it's going to be pretty difficult to see for you. Now I can take it off because there's basically no liquid for the beads to be resuspended in. But you basically have to take a closer look at the bead pellet. I think it's pretty impossible to see from there. But if the bead pellet is glossy, means that it still has ethanol, so you still have to wait. However, if the bead pellet is not glossy anymore, and it's, let's say, like a matte brown, that it means that it's basically the perfect timing to resuspend it, okay? If you start seeing cracks, that's how they're called in the jargon, in the bead pellet, that means that you over dried your bead pellet, okay? You start seeing few cracks, which means that it's about time for bead resuspension, okay? So I'm adding to my tube 25.5 microliters of the Elution buffer. The Elution buffer that I'm using right now, it's EV buffer from Kaigen, which is basically 10 millimolar tris pH 8. So you can do it in the lab. So I'm just going to resuspend it. And as you can see, it was pretty easy to resuspend. And there you go. Okay, so we suspended my beads. Let's check the other one. So the other one is still not ready. I'm looking at the bead pellet, no cracks. Now it's not glossy anymore. Okay, so I can add my resuspension buffer. I think it's pretty difficult to see because the tube itself, it's kind of like a glossy, so it's pretty difficult for you to see that the bead pellet is not. Okay, but trust me. Okay, so I'm trying to do like a close-up. I'm adding the Elution buffer. In this case, I'm adding the Elution buffer directly on the beads, okay? So this is the only time where I can pipette on the beads themselves. And there you go. And so you start seeing the bead pellet is resuspended in our Elution solution, all right? That's it. I usually prefer to vortex it for a few seconds just to make sure everything is very well resuspended. And now I can incubate at room temperature for 3 minutes. After the room temperature incubation, it's almost over. I'm just gonna spin my tubes down to collect all the resuspended beads at the bottom of the tube. One and two. I'm just gonna place my tubes on the magnet, and differently from the first incubation of the tubes on the magnet, this time the separation is almost instantaneous. So it's probably taking like 30 seconds or so. Why is that? Because now the media where the beads are in is not viscous. It's just basically a water aqueous solution, right? So before it was, very viscous. So it took like a little bit of time. And as you can see that the bead pellet is already separated from the rest. Same here. So we are basically ready to go. I'm gonna transfer. So I've resuspended my samples in 25.5 microliters of EB buffer. I'm gonna remove 23.5. I basically lose 2 microliters of my sample in the tubes, but in this case, I don't want any bead carryover, which might interfere with the next PCR. So I'm just willing to lose 2 microliters not to have interference. I'm transferring 23.5 microliters into a new PCR tubes, and then we're gonna perform the final PCR. Okay, as you can see, no beads carry over. Okay, in this case, this is not supernatant to be discarded. This is just our purified DNA library. Okay, so I'm just gonna pipette this in here. Okay, you can still see the bead pellet here and here. And we can discard these tubes because they don't have anything bound to it. Okay, so you can also see the different bead pellet size between sample B and sample A. All right, so we are now ready to perform the PCR, and we have our purified DNA after adapter ligation. We are adding the PCR primers. It's a 1.5 microliters each. Okay, and then we are adding the PCR master mix, 25 microliters each. Closing the tubes. Vortexing, spinning down, and our volume for the PCR is 50 microliters. Remember, 23.5 microliters from the purified adapter ligated DNA, 1.5 microliters of primers and 25 microliters of the PCR master mix. This in here, and the program is already set. We're doing 5 cycles of PCR, so it's quite quick. It's about 15 minutes, and then once it's done, we come back, we perform the final purification, and then we are ready to load it on the TapeStation. Our PCR is almost concluded. Once our samples reach the temperature of about 20-25 degrees, I can take them out and perform the purification right away. Again, you can purify your samples both in PCR tubes or in 1.5 ml tubes. I'm going to try to show you again one more time in 1.5 ml tubes how the purification works. Hopefully you'll see it a little bit better. And then after purification, again, we're going to quantify the samples using the Qubit and also load the samples on the TapeStation. One more time, all the steps for the purification, they have to be performed at room temperature. So that's why we are incubating, 1 minute just to make sure everything is at room temperature. In the meantime, I get my Ampure beads ready. So they're still the same beads as before. Again, we are purifying our sample with 2 different methods. One method, it's a normal purification, requires 0.9 volumes of beads compared to the DNA. So if our PCR product is 50 microliters, 0.9 volumes are 45 microliters, while the other sample that we're trying to push, we're going to purify it with 1.8 volumes of Ampure beads. So it's the same as before. We have 50 microliters now of PCR, and then we're adding 90 microliters of Ampure beads. Okay? So let's get started. And incubate for 5 minutes. Okay, so our incubation time is almost over. I'm gonna spin down these tubes, and again, I'm gonna transfer them into 1.5 ml tubes so you can see the purification a little bit better. And now we can incubate for 5 minutes to have the beads separated from the supernatant. Okay, so our incubation time is almost over. As you can see, the beads are pelleted against the magnet. Do the same view as we did before. As you can see here, you see the pellet only on the bottom part. And again, this is where the magnet is actually. So everything makes sense. So I'm just gonna remove the supernatant. The first removal of the supernatant, again, I just wanna remind you, you may wanna do it with a pipette and not to basically retain beads. So the beads now have bound the DNA of your interest, okay? So you don't wanna discard the beads with DNA that you wanna purify. So I'm just gonna remove 133 microliters for the sample B. Okay, so I'm going on the opposite side as the beads. And again, I just wanna check if I don't have any brown color on the tip. That will signify that I have some beads that I'm gonna discard. If this happens, actually you can just put the supernatant back in your tube and then just wait like a few minutes for the separation. And then maybe instead of removing, like in this case, 133, you remove 125 or something like that, okay? So I'm just gonna discard my tip with my supernatant. Now I'm gonna discard the supernatant for sample 1 or sample A. And we have 50 plus 45, so we have 95 microliters. I'm gonna remove 87 microliters in this case and see if that is good enough. So again, just want to show you, I'm just going in with my tip on the opposite side. Maybe this way is easier. Move it quickly. I don't know if it's easier to see, and just going in on the opposite side of the tube, and I'm removing the Supernatant. Okay. Again, you just want to double check no brownish tip, so you can just discard it. And now we proceed with the ethanol washes. Okay. Again, we use 300 as the volume for the ethanol washes. We're using ethanol 80%. Okay. So I'm just adding the ethanol on the opposite side of the beads. I just don't want to disturb the bead pellet. Now we wait like 30 seconds for the wash, and we can remove the supernatant using the vacuum. Okay, same as before. All right, 30 seconds in. Just going with the vacuum. I'm going on the opposite side of the beads. You don't want to put the vacuum on top of the beads. Your experiment will be gone. So I'm just going on the opposite side, and there you go. First removal, and second removal. Okay, going on the opposite side, go all the way in, and there you go. We add the ethanol once more. And now we wait 30 seconds. I'm just going to show you. Again, the bead pellet, it's there. The reason why we're removing the ethanol with the vacuum instead of the manual pipette, it's because now it's more difficult for the beads to be resuspended in ethanol solution. So I can use a stronger removal method. It's much faster, much cleaner. Going in, opposite side. Going in, opposite side, and removed. Okay. So now we wait a couple of minutes, and then we go back in, remove the excess ethanol that it's being collected at the bottom of the tube, and we look for droplets of ethanol on the top of the tube just to make sure there's no ethanol carryover. Once the beads are dry, not too dry to have a lot of cracks, and not glossy, which means that they are still wet. At that point, we can resuspend using the EV buffer in 22 microliters, and we are going to recover 20. So remember, every time I recover from the Ampure beads, I usually prefer to leave like a 1 or 2 microliters in, not to have beads carry over. Okay? All right, 2 minutes. Expired, almost expired. So I'm just going in, removing the rest of the ethanol. I'm looking for some other droplets. Sample is good. And then I'm going in on the other sample and remove the ethanol at the bottom of the tube, and then looking for some other droplets. Okay. Now we set the timer for 3 or 4 minutes, and then check frequently that our samples are whether our samples are dry or not. All right, so let's check. Time is almost over. I'm seeing that the sample A is ready. I start the first few cracks appear. So it's perfect timing to just add the 22 microliters of elution buffer. Now it's pipetting on top of the beads to resuspend them. And there you go. Again, I prefer to Vortex my samples to mix them better. Let's check the other sample first. Maybe 30 more seconds, and then we can resuspend it. So in the meantime, I'm just gonna vortex my sample. Make sure everything is very well resuspended. And now, it's time to resuspend. I don't know if you can see it. It's probably very difficult to see just because the tube is glossy as well. But believe me, these beads are dry. Okay, so I'm just gonna add 22 microliters of EV buffer for this sample as well. Again, pipetting on the top of the beads. And I'm gonna vortex one more time. And now we wait 3 more minutes for the resuspension. Okay, so the time is almost over. I'm gonna spin those tubes down to collect all the liquid at the bottom of the tube, and then we can put them back on the magnet for the final separation. If you remember, the final separation occurs very rapidly. So in a matter of like 30 seconds, the beads are already pelleted against the magnet. So I am able to transfer my eluted DNA into new tubes, and then we are going to check the size distribution of the TapeStation and the concentration with the Qubit instrument. Now I'm setting my pipette to 20 microliters. Sample number 1, I just go in, opposite side of the beads, and there you go. I've taken my Supernatant. Now I pipette that into my samples tube for the final libraries, 1 and 2. Okay, so our libraries are now ready. Hopefully we will see what we expected to. So one library, which is like prepared using the regular protocol, that has only the smear of the library with a size of around 350, 400 base pairs, and the other library, which has the library smear plus primer dimers and adapter dimers. I want to remember you, primer dimers are usually between 60 to 80 base pairs in size, while the adapter dimers are usually between 130 to 160 base pairs, depending on the type of adapters that you used. They might run a little bit differently. So let's go to the TapeStation first, and then we can go back and check the qubit concentration using the qubit fluorometer. Okay, so it's 3:33. Let's take a break of 7 minutes, and then I'm just gonna take a quick break of a couple of minutes, and then I'll be back and then answer 2 questions. If you have, please post them in the Google spreadsheet. Okay, I'll see you all at 3:40. Okay, so it's 3:40. We're gonna start back on part 3. So this part is basically we are reusing the TapeStation. Just this time we are checking the final library instead of checking the initial quality of the RNA that we did at the very beginning of this class. If you remember from day 1, these were 3 different types of library preparation. Okay, so if you remember, this was A,
B, or C. So we're trying to see our two libraries, which category they belong to, okay? So our goal is actually to have one of the two libraries, the one that use the standard protocol, to look like C, and the other library to look like B, okay? So to contain adapter dimers and maybe some primer dimers as in A, okay? So that's our goal. Let's see how we did in the prep. It's gonna take 18 minutes. So after that, I'm gonna pause for maybe a couple of minutes to just check for questions, and then if there are no questions, we can just go ahead and look at the final video of the Qubit, and then we're gonna talk about how to submit the library. And at that point, I'm just gonna talk live. So we are in the final stretch, finally, of this workshop. I know it's very boring to just watch and not doing, not pipetting, but you know, there's nothing else we could have done. Okay. So let's go to the next video. Just give me a few moments to get it on. Oops. Why is this not working? Sorry, just a few seconds. I don't know why Zoom is not giving me the option to share. Yeah. Okay, again, I'm just gonna play for a few seconds, and then I'm just gonna ask you if you are able to see video and audio.
Okay, so now we're gonna use the TapeStation to visualize our final libraries. Okay, so the two libraries, awesome. Remember, one of them was prepared according to the standard protocol. The other one was artificially pushed to contain adapter dimers. Let's see if we did a good/bad job of retaining the adapter dimers, okay, which I remember you, it's a very bad contaminant. So what we're gonna do right now, it's we're gonna use the D1000 screen tape regular sensitivity. I'm just gonna pipette the RNA. I'm just going to pipette the DNA sample buffer. In this case, the protocol requires 3 microliters of the sample buffer for each sample, and 1 microliter of my sample. Okay. One other good feature of the TapeStation is that it actually requires very little material to begin with. As you've noticed, both for DNA and RNA, they're just asking for 1 microliter. And especially for the final library, you don't have too much material to work with. So it's always nice when you don't have to load a ton of material on a gel. So if you want to visualize actually a smear, because the final libraries are going to look like a smear, and you'll see in a minute, on a gel, if you want to visualize a smear on an ethidium bromide agarose gel, you need to load about, I usually prefer to load about 70 to 100 nanograms, which is quite a lot for final libraries, okay? And we discussed it yesterday, that if you have a contamination of adapter dimers of 1%, that's basically borderline to be sequenced. Otherwise, you're going to have a huge contamination in your final data. So if you want to have a 1%, being able to detect 1% of adapters, and the, let's say, the lower limit of detection on the ethidium bromide gel is a band of 10 nanograms, okay? So if you do like 1%, if 10 nanograms are 1%, means that you have to load 1 microgram of the final library to be able to see it on an agarose gel. which means that you have to do a lot of PCR cycles, which means you will introduce a lot of PCR duplicates in your final reads, okay? So that's why the TapeStation or the Bioanalyzer or other systems that, are actually designed for this purpose are really nice, okay? So I've added 3 microliters of my sample buffer, 1 microliter of my DNA final libraries, and then I'm just gonna Vortex. The actual protocol calls for 1 minute on the multi-vortexer that is actually next to the TapeStation. I usually prefer, and I find it more appropriate and more effective to just vortex few seconds on a regular vortexer manually. Of course, if you have 96 samples, then I'm gonna tell you, okay, just follow the protocol, just vortex on the multi-vortexer, okay? So I'm just gonna spin it down. This time, differently from the RNA, there is no incubation step, no ice incubation. So we're basically ready to go with our samples, our tips, and our tape. All right. Okay, so here we are on the other side next to the TapeStation. So again, we're just gonna open the TapeStation, load our samples. Again, you can place your samples anywhere. Okay, so I'm just gonna place my samples here, same positions, A1, B1. And then again, I'm gonna add 16 tips to my run. Sorry, yeah, 16 tips to my run. There you go. And I'm gonna put the tape in, okay? Again, orientation, the QR barcode goes on the bottom facing the laptop, okay? So I'm just gonna place it here and I'm ready to get started. Okay, so I'm just gonna close this one and we're gonna transfer onto the TapeStation laptop. Okay, so here we are. Let's open the TapeStation controller. It's going to connect to the machine. I already loaded the machine. Just remember to remove the caps from it. It's going to give me an alert that I have my tape is expired. That's true. I used 4. Let's see if it's true. Yes, I have 4 dark gray lanes in here, and I have 12 light blue lanes to be used. Okay. So as you remember, we put our samples here, top left. So position A1, B1. And automatically the machine thinks that you are using a ladder. We are actually not using the ladder, so I'm just gonna click on no ladder, and I can put my sample names 1 and 2, and everything else is in. 16 tips in the tips holder, so we're good to go. It requires only 2 here, and then it highlights the lanes that are going to be used. So I'm just gonna press on start and save it under the collaboratory. So we're going to call it workshop 16 DNA. All right. So I'm just going to click on save, and then same as before, click on OK, and then the run will go ahead. It's not going to be a long run, but I'm going to pause the recording here, and we're coming back later after the run is done. Okay, here we are with the results of our DNA TapeStation on the final libraries. And as you can see, we have 2 samples. We don't have any sizing information, as you can see here on the tip table. So the first thing we're going to do is to insert the ladder. So now the electronic ladder appears here. Again, you see the green bar at the bottom, which is the lower marker. And another difference between the DNA and the RNA is that with the DNA, you have this purple bar at the top, and this is actually the upper marker, okay? So in the case of the RNA, you only have the lower marker. In case of the DNA, you have both lower marker and upper marker, okay? And they are indicated with the green for the lower and purple with the upper marker, okay? If you remember, second thing that you usually do is to scale to sample. So you're just like visually normalizing your results. It's much more, much easier to see, in my opinion. So this is the gel image. You can see that the two samples are actually different. Okay, so A1 has just one smear centered around, let's say, like 350, 400 base pairs, while the sample B or sample 2 has a peak that it's slightly lower, or a smear that is slightly smaller in size, or like 350, 330. And also it has another peak right here around 130, 140. But let's take a look, a deeper look using the electropherogram, okay? So I'm just gonna click on electropherogram and I'll see the fake electronic ladder that I used. And then I have my sample here, the first sample, okay? As you can see, the lower and upper marker are present. And then you have this smear right here, which is centered around like 380 base pairs. This is actually, just want to mention, this is not the average size. We're going to calculate the average size using a separate procedure. But as you can see, you have just one smear, so no peaks in this region, which is, if you remember correctly, the region of the primer dimers, which is usually between like 60 to 80 base pairs, and the adapter dimers, which is usually between 140, 150 base pairs, okay? So if I go to the other sample, now I'm starting to see multiple peaks, okay? So I have the lower and upper marker. I have my smear of interest, which is, again, smaller, centered in a smaller size, okay? So 330. And but you have more peaks, okay? So you have a peak around 70 base pairs, and you have a peak around 130 base pairs, okay? So these are actually primer dimers at 70 base pairs and adapter dimers at 130 base pairs, okay? So we actually were able to push by increasing the amount of AMPure beads for this specific, we did two things actually. We increased the amount of AMPure beads, and we actually increased the amount of adapters used in this particular level preparation. We were able to induce the presence of adapter dimers and primer dimers, okay? If you want to quantify this, you can just go to the region table, okay? Same as before, you can do it manually if you want. You can do basically create a region, or what you can do is you can delete all region if you did it manually. You can do it automatically, so file, options, and then I already preset 50 to 150, let's call it dimers. It's both adapter dimers and primer dimers. And 180 to 700, we can actually make it a little bit bigger to 800, our libraries. I'm showing how to name the regions. So I'm just gonna apply file, click on OK. As you can see on sample A, you don't have any here. So it's like 0.16% total. That's basically background. And if you look closely, that's actually a tail of the lower marker that it shows up here. And then you have most of your sample that it's between 180 and 800 base pairs, which is good, a great library. Okay, so no need for further purification. If you go to the other sample, sample B1, then you have the amount of your dimers region. It's close to 9%, which is super bad. If you remember, the threshold that we usually set is 1%. So if it's below 1%, we consider it quite good. If it's above 1%, then it requires a further purification, okay? And then in this region right here, you have your library, which is still high, but you have too many dimers, so you need an additional purification. How you do that? You perform a AMPure beads or spry beads purification I usually prefer to do, in this case, a ratio of 1 to 0.8, so 0.8 volumes of the beads. So I usually bring the library up to 50 microliters, and then I add 40 microliters of the Ampule beads. So I have 50, which is 1, and 0.8, which is 40 microliters of beads. And then I perform exactly the same steps that we've seen before. Okay. What else? What other information you have from the region table? It's the average size. Okay, so this is a really important information that you have. In this case, the average size is 379. Okay, we will need this information to calculate the molarity of our samples. Okay, you'll see that the concentration will be variable from the qubit concentration that we have. Usually, you have twice as much material measured by the qubit compared to the TapeStation. Okay, so that's why we don't usually use the TapeStation to quantify our final libraries. You have two options. You can use the qubit, and usually it works fine on our end. Or if you want to have like a more accurate concentration, because it's measuring only the amplifiable DNA, you should use a Qpcr-based method. I'm not presenting this one here. It's going to take too long. But again, I just want to let you know that these two are two viable options to quantify the concentration of the DNA. Okay. So again, we are going to create a report. We are going to save it, and then we're going to save it on the desktop. I'm just going to show you how it looks like. I'm just going to save it as a Word document. The PDF gives us some errors. Just save it, and this one is done. I'm going to close it, save, close this one, and it should be this one, DNA. Just going to open it, and then again you have the first page, which is like a summary page, and then you have one page per sample. So in this case, you have the electronic ladder that we didn't run, and then we have sample 1. And you have the electropherogram, you have the region table, so the region electropherogram and the region table, which gives you the average size and all the information about the concentration, the amount of dimers, and also B1, which has high amount of dimers. Okay. So that's it. So you have your Word document with all the information. And we just close it. And next thing that we have to do is to clean up the TapeStation for the next user. Again, just want to show you one more time. So just gonna stop sharing here. I'm just going to go back here. We are keeping this tape because we still have some unused lanes. So I'm just going to put it back at 4 degrees. Also, I'm going to collect the unused tips, put them back in the tip holder, and then I'm just going to empty. I don't know if you can see it. You have 2 tips here, the ones that we've used. I'm just going to empty in the trash, and now it's clear. Okay, so no tips whatsoever. And then our samples have been run already, so we can just discard it. I just want to mention, you see the sample buffer now, it's greenish blue, while the RNA buffer that we used before was yellowish slash colorless. Okay, so this is telling me to remind you that each application has its own buffer, its own ladder, its own tape. Okay, so don't try to run a RNA screen tape with the DNA buffer, and vice versa. Okay. Also, within the RNA sample buffer from the regular sensitivity is different from the high sensitivity. Actually, the lower marker is lower or upper marker, depending on which application you're using. It's used for quantification as well, not only for running. So you just want to make sure you understand that you need to use exactly the buffer that is specific to that application. Okay, and with this, the TapeStation session is concluded. and I'm waiting for you to ask me more questions.
Okay, so just a couple of minutes if you have any questions. I don't see any at the moment. If you don't have any, at 4:02, we're gonna start the last part of the workshop, and I'm gonna just quickly present the video that we're gonna look at, and that's the last video that we have. It's the Qubit video, and then we are gonna discuss how to submit the libraries, okay? So just gonna give you a couple more minutes while I'm pulling the video, and that's it. Looks like no questions. Same as before, after a few seconds, if you can just type in yes and no if you can hear anything at all. And then if everything looks good, you can just go ahead. This is like a much shorter video, it's like 12 minutes only. Bear with me. And then we're gonna finish up pretty soon, okay?
Okay, welcome back for the Qubit measurement video. So this machine is actually the Qubit. It's a fluorimeter, and it measures the concentration of the nucleic acid that you want, also actually proteins. After you after you've incubated your sample with a dye that is specific for the molecule you want to measure. Okay, so what do we need for the Qubit measurements? Of course, we need the Qubit itself. We need specific tubes, and in this case, I have 4 empty tubes. 2 of them will be for the standards, and 2 of them will be for our samples. On the other side, I have our standard. They come with the Qubit, and I have a empty tube for the working solution that we're gonna make relatively soon. I have the buffer, which is basically needed for the preparation of the working solution. And then I have a box of the Qubit dyes. They are stored in the dark just because they're light sensitive. So I'm gonna take them out only for the pipetting step, and then I'm just gonna put them back here. Both the buffer and the Qubit dyes are stored at room temperature, while the standards are stored at 4 degrees. And so for precise pipetting, I would suggest you take them out 10 to 15 minutes before performing the measurement, okay? As I told you before, you can run different assays using the Qubit. What we are using today, it's called double-stranded DNA PR, which stands for broad range. And each assay has its own specific reagents. Standards are specific for DS DNA BR. The buffer is specific for double-stranded DNA BR. And also the dye that we're gonna use later is specific for double-stranded DNA BR, okay? If you wanna run other measurement assays, such as DNA high sensitivity, then it's gonna have its own set of standards, buffer, and dye. The other assays that are available with the Qubit are RNA of regular sensitivity and high sensitivity. Also DNA single-stranded. The assay works that you prepare a working solution that it's composed of 199 microliters of the double-stranded DNA BR buffer in this case, and 1 microliter of the dye. The sum, as you can see, it's 200, and that's exactly the final volume of the assay that we are gonna perform for each of our samples. how many samples do I have to prepare the master mix for? We have 2 samples to be measured. We have 2 standards, which is 4, and then we're gonna prepare 1 extra just to account for pipetting errors, okay? So 199 we can approximate to 200. So it's 200 times 5, so it's 1 ml. And then we're gonna pipette 1 microliter of the qubit dyes into this empty tube of the working solution. After we're done, we're gonna pipette our working solution into our tubes, and then we're adding either the standards or 2 microliters of our samples. I told you before, the final volume of the assay is gonna be 200. So since we are pipetting 2 microliters of our samples into these tubes, we have to add 198 microliters of our working solution. So it's 2 plus 198, it's equal to 200. While for the standards, the protocol calls for 10 microliters of the standards from the tubes. So it's 200 minus 10. So we're adding 190 microliters, 190, of the working solution into each of the standard tubes. And then we are ready to add our standards. All right. Okay, so I've added my buffer. Now we add our dye. So this is the Qubit double-stranded DNA BR dye. It's called reagent. It's basically a dye. It's bright red. So I'm just gonna vortex it to mix it, spin it down, and then I'm ready to pipette 5 microliters for our assay. Okay, so this is our working solution. Vortex it to mix, spin down. And there you go, ready to go. So again, we are measuring 2 microliters of our DNA, and we are adding 10 microliters of our standard DNA in there, which means that we're adding 198 in each of the tubes for our samples and 190 for our standards in order to have a 200 microliters volume for the qubit assay. And as you can see, I have extra because I prepared for 1 extra sample. Just gonna pipette the standards. We have standard 1. So standard 1 is basically blank. So there's no DNA, just the TE buffer. So it's gonna be our blank control. And then standard 2 is actually a defined concentration of DNA. Now we are pipetting 2 microliters of each of our DNA in there. Okay, so in each tube you have buffer, dye, and your sample, okay? Or your standards, okay? So what I'm gonna do is I'm gonna briefly vortex them, and then we're gonna spin them down. Just to collect all the liquid at the bottom of the tube. Now we incubate for 3 minutes at room temperature to have basically the dye bind the DNA, and then we can measure our samples with the qubit. Okay, so the incubation is almost done. I just want to show you the qubit, how it looks. So as soon as you plug that in, you have all the different assays that you can perform. Today we're gonna push DNA, and then you have other assays you can choose from. It's double-stranded DNA high sensitivity, top left, double-stranded DNA broad range, top right, and then you have single-stranded DNA. We're gonna push double-stranded DNA broad range. That's the assay that we're gonna perform. So the machine is asking us, do you want to run new standards? The answer is yes. I usually do every single time. And then we are ready to insert our standard number 1. And then we're going to press read, which is here at the bottom. We're going to repeat the operation with standard number 2. And then we're actually going to reread standard number 2 as a sample to see its concentration. Okay. After that, we're going to check our samples. All right. Time is up. Now we can measure our standards. So I'm just going to put my standard back in here, close it, click on read, and the standard 1 is reading. All right. Now we insert standard 2 so we can create like a standard curve, read. And now we have our standard curve. I don't know if you can see it, probably not. But we have a standard curve. So now we're going to reread standard 1 as if it were a assay tube. And as you can see, you have sample too low. Of course, it's only buffer, okay? So the concentration is too low to be detected. And then we're going to insert standard 2 and then reread it as if it were a assay tube. And now you have a very high concentration. So it's about 5, okay? So microgram per ml, okay? So if you want to calculate the concentration of your actual sample, so this is the concentration that appears here on the screen is actually the concentration of your tube, your diluted tube. Remember, we've added 10 microliters in 200 microliters of assay volume. So now we can click on calculate stock concentration, which is at the bottom right here, and then tell the machine, okay, so what is my what is the volume of the sample that I used? And I used 10 microliters, so I set it to 10 microliters here. And then you see the concentration, which is close to 100 nanograms per microliter, which is what it's supposed to be, okay? So now that I have everything set up and I know it's working, I add sample A, and then just gonna read my next sample. I use 2 microliters in here, so I'm just gonna calculate the stock concentration and then set it to 2 microliters here. And the concentration of the stock tube that, basically the concentration of what I have in my purified PCR tube, it says it's 41.9 nanograms per microliter. And then I'm gonna measure sample B. I'm gonna read next sample. And it says that the concentration, once I calculate the stock, it's 43.3 microliters, okay? So this is our final concentration measure with the qubit. You notice that the concentration is different from the TapeStation concentration, more or less in a ratio that I told you you would expect. But we think the qubit is more accurate, and we've had like a lot of experience with the qubit to submit libraries, and it always works fine. So we usually use the average size of the TapeStation and the qubit concentration for our final libraries for the calculation of the molarity. And we're going to see the calculation of the molarity in a second.
Okay. Can you guys hear me, and can you see the presentation now? Okay. So we've talked about the qubit. So we use the qubit to calculate the concentration. I just want to stress out one thing. It's also possible to use the Qpcr method to check the concentration of your final library, okay? These are just 2 different methods. If you have 96 libraries you want to measure the concentration, the Qpcr is going to be faster. In general, so let's say in general, the Qpcr is actually more accurate than the qubit because it's measuring only the amplifiable DNA. But if you have a good library, most of the times the qubit and the Qpcr are quite similar. Okay, so you can either you can use either or. Okay, basically the information that we have now are 2, right? So we have the concentration, and then we use the qubit to measure the concentration. We also have the average size, okay? And we got the average size from the TapeStation, okay? And with these two information, now we can calculate the molarity, which is the basically what the sequencing core is asking you for each of your libraries before submission, okay? So how you do that? There are two possibilities. Either you go to this website, okay? And you basically input, your average size here up top. Just make sure it's in kilobases, it's not in base pairs, so that's why it's 0.3 in this case. And then you put your concentration here. In this case, the picture I took was 21 nanograms per microliter based on the qubit concentration. And automatically the program is giving you the molarity at the bottom. Okay, so in this case it was 107 nanomolar. Okay, the other option is you use a formula. Okay, and it's this one. So you're basically using the concentration from the qubit in nanogram per microliter, and then you multiply by 10 to the 6th, and then you divide everything by 660 times the average size in base pairs. Okay, so the number that it will pop up, it's the concentration in nanomolar. Okay, so that's the formula that we are using usually. Okay, so let's make some examples. So this is the table that we got out of both the qubit and the TapeStation. Okay, so this is the tube. So we have the two standards here up top, and this is the concentration that you get, but we don't care about we're not submitting those guys for sequencing, so we're not calculating anything. We have the DNA1, so the first library that we prepared. We have its concentration, 41.9, and the average size, 426 base pairs. Okay, so the calculation is giving me 149 nanomolar. Okay, so I'm just giving you a couple of minutes. You can try to calculate the molarity of DNA sample 2 or B based on these values, and then write down the answer in the chat, please. Okay, so you can use this formula right here to calculate the molarity. So just want to make sure you understand how to do this. Okay, I'm just going to give you a couple of minutes. Okay, sounds good. Yeah. So if you use the formula, you're gonna get 173. If you use the link that I gave you in the previous slide, it's possible that it's slightly different. They're like I think like few approximations. So for example, like this is an approximation of the molecular weight of the base pair. But in general, it's very similar. Okay, so yeah, the answer correct is 173. Okay, there you go. All right, so what are we doing next? So we have the concentration of the molarity concentration of both our libraries. So what I usually do is I prepare a dilution. Okay, so let's look up here, you have your final libraries. Okay, so we have done this. We've measured with the qubit. We have the averages from TapeStation. Sorry, we've calculated the nanomolar concentration. So what I usually do is I dilute every library that I have to 10 nanomolar. Okay, so there's different strategies to do that. I think the easiest to understand it's this methodology. So I dilute every sample to 10 nanomolar, and then I mix an equal volume of each sample that is already 10 nanomolar into the same final tube. Okay, so in the picture here I had 6 libraries. In the case that in our example we only have 2, but you know, you can have as many as you want, like 96. It's just like, you know, it's going to take a long time, but that's how I usually do it. And how you prepare a 10 nanomolar dilution, it's pretty easy. So I usually prefer to pipette 2 microliters of my final library. And the concentration, the amount in microliters of the sequencing solution. So the sequencing solution is the solution that you have to mix your final library with before submission. And it's very simple. It's just like the same as EB buffer. So trace 10 millimolar pH 8 with 0.1% of twin. And so this is a formula if you want to calculate how much of the sequencing solution you have to put with your 2 microliters of the final library to create a 10 nanomolar dilution. Okay, so for example, for sample 1, we are using this formula. So this is the nanomolar concentration. So it's 2 times 149 divided by 10 minus 2, because this is the volume that we are already using for our final library. It's a 27.8. Okay. So what I'm going to do at the end, it's 2 microliters of my final library and 27.8 microliters of my sequencing solution. Okay. So I'm just going to give you a couple of minutes to calculate how much volume of the sequencing solution you need for sample 2. Remember the correct answer for the concentration was 173 nanomolar. Okay, so just write in the chat what's the solution in your case. Okay, sounds good. I hope you didn't go to the next slide to check the result. You did the calculation. It's, you know, it's just for you to just understand how it works. So that's correct. So the correct answer is 32.6. And so basically at the end of this dilution, everything is a 10 nanomolar. Okay, and so you can mix these 10 nanomolar solutions with the same volume, which means that you're going to put the same number of molecules in the final tube for each library. The concentration of the final tube, it's still 10 nanomolar, okay? Because it's just measuring the concentration of the total DNA that you have in. The concentration of each of your library in the final tube will be 10 nanomolar divided by the number of libraries that you put in. So if it's 2, like in the case of our example, each library will be 5 nanomolar, okay? Even if the final concentration will still be 10 nanomolar, okay? If you have more, like for example, 5 libraries, it's gonna be 2 nanomolar and so on, okay? There's also another strategy to pool the samples. It's more complicated, but Illumina, for example, has on this in this link has a pooling calculator. So if you basically add the concentration of each of your libraries, it will tell you exactly how many microliters of each library to put into the final tube without going into this intermediate dilution step. I prefer to do the dilution step because I have controls over the volumes that I'm pipetting. Sometimes the Illumina pooling calculator is asking you to pipette 0.3, 0.4, 0.5 microliters, which is not a very accurate pipetting step. And also I usually have extra of my 10 nanomolar diluted libraries in case the sequencing core wants to resequence, or I want to resequence with more depth my libraries. So I still have the 10 nanomolar dilution, so I can just quickly remix my samples and resubmit them, okay? And this is the final answer, and that's it. So I'm just gonna quickly check for questions. This is your last chance of asking me questions about anything. And then there's gonna be one slide, and then I believe is gonna be final quiz time, okay? So let me check quickly. I don't see any, so I'm just giving you like 1 minute in case you wanna ask something. But then if nothing happens, we're just gonna go directly into the last slide, which is the final quiz, okay? While I wait, I don't see anyone writing anything in here. Resources that the QC Bio offers that are regarding my workshop, or my fellowship, is I can prepare a library preparation for free as long as you provide the reagents and consumables. So the kits and the pipette tips and so on. It's going to be a collaboration, okay? So you contact my PI, which is also the director of the QC Bio, and start a collaboration, okay? I have office hours anytime, so just write me an email if you want to consult me about, I want to do a library preparation. What's the input that you recommend using? Is it difficult? And so on and so forth. Also, I have a library preparation calendar. And if I remember, I'm basically adding the library preparation that I'm preparing at the moment. So if you want to tag along and learn, you can always do that. It's just like, it has to be according to my schedule. So, I usually cannot accommodate, your schedule very much. Also, we have a liquid handler in the lab, and you are welcome to use it. I have to give you training on how to use the liquid handler. And if you want to, we have some applications that are already scripted in the machine. For example, RNA-seq, which is very popular, or some basic DNA sequencing methods. So if you want to use the machine because you have tons of samples and you want to waste your life over library preparation, just talk to me, and then we can program your protocol on the liquid handler, and then you can perform the library preparation in using the robot, okay? Also, I don't know if I mentioned in the video, the TapeStation is available for everyone at UCLA campus. If you want to use it, of course, you have to buy your own reagents and consumables, but just write me an email, and then I'll send you all the reagents that you need with the catalog numbers and how to schedule a run, okay? I have to update these slides. We also have a sonicator that you are welcome to use if you want to fragment your samples. It's a Bioraptor Pico. The only thing you have to buy is are the tubes, and that's it. Again, if you want to use it, just send me an email. I'll add you to the Google Calendar that we have, so you can just schedule your time, and that's it. Okay, so ready for the final quiz. This final quiz is really important. Okay, it will grade your class, and also it will take attendance for today, okay? So I'm just gonna share the link of the final quiz in a second in the chat, and good luck, everyone. If you have questions, I'm gonna be here for the next 20 minutes, and otherwise send me an email, okay? Good luck, everyone. Okay, the quiz will stop at 4:50. Okay, 4:50 p.m. If you want few more minutes, just write in the chat. Okay.